Welcome to the StudyNeuro session on the pituitary gland, one of the most fascinating and vital endocrine organs in the body. Today, we're going to explore not just its anatomy, but also the specialized cells that produce hormones and how these hormones control key functions like growth, metabolism, reproduction, and blood pressure. We'll start by locating the pituitary within the skull and understanding its relationships to nearby structures, knowledge that's essential for both clinical practice and surgical planning. Then, we'll break down the gland into its anterior, posterior, and intermediate lobes, uncovering the unique cells in each and the hormones they secrete. Along the way, you'll see how the hypothalamus orchestrates hormone release, ensuring the body stays in balance, and why disruptions here can lead to conditions like pituitary adenomas or systemic endocrine disorders. So get ready to connect anatomy, physiology, and clinical insight in a way that makes the pituitary both understandable and highly relevant to patient care. And remember, for more neuroscience and clinical updates, follow StudyNeuro across YouTube, Facebook, TikTok, Instagram, and Twitter at StudyNeuro. The pituitary gland is a small, pea-sized endocrine organ located at the base of the skull, sitting within a bony depression called the sella turcica of the sphenoid bone. It is connected to the hypothalamus by a thin stalk known as the infundibulum, which allows the brain to control its hormone release. Superiorly, it is covered by a dural fold called the diaphragma sellae, which has a small opening for the stalk. Despite its tiny size, roughly half a gram and measuring about 12 by 8 millimeters, the pituitary is often called the master gland because it regulates critical body functions, including growth, reproduction, metabolism, and blood pressure. The pituitary gland controls key body functions through its hormones. Thyroid-stimulating hormone regulates metabolism, growth hormone promotes growth, and adrenocorticotropic hormone supports the stress response. Vasopressin maintains blood pressure and water balance, while follicle-stimulating and luteinizing hormones control reproduction. Prolactin drives lactation, oxytocin stimulates labor and milk ejection, and melanocyte-stimulating hormone influences pigmentation. Together, these hormones coordinate growth, metabolism, reproduction, and homeostasis. The pituitary gland is divided into three main parts, each with distinct roles. The largest portion, the anterior pituitary or adenohypophysis, makes up about 80% of the gland and is composed of true glandular tissue that produces a variety of essential hormones. Behind it lies the posterior pituitary, also called the neurohypophysis or pars nervosa, which is made of neural tissue and primarily serves as a storage and release site for hormones produced by the hypothalamus. Sandwiched between these two is a very thin layer known as the intermediate lobe, or pars intermedia, which is relatively small in humans but contributes to hormone regulation. Together, these divisions allow the pituitary to act as the body's master regulator, coordinating a wide range of endocrine functions. The pituitary gland sits in a bony depression called the sella turcica, which has distinct boundaries. At the front, it's limited by the tuberculum sellae, and at the back by the dorsum sellae. Below, the floor is formed by the sphenoid sinus, an important landmark for transsphenoidal surgery. Above, the gland lies just beneath the optic chiasm, so any upward growth of a tumor can compress the optic nerves and affect vision. On either side are the cavernous sinuses, which house cranial nerves 3, 4, and 6, as well as branches of the fifth nerve, making lateral extension clinically significant. Inferiorly, it relates to the sphenoid sinus and nasopharynx, and in rare cases, tumors can erode the surrounding bone. Posteriorly, the pituitary gland is related to the dorsum sellae. The pituitary stalk, or infundibulum, passes through the diaphragma celli, connecting the gland directly to the hypothalamus and allowing precise hormonal control. The anterior pituitary, also called the adenohypophysis, is itself divided into three main regions. The largest portion, the pars distalis, forms the functional bulk of the gland and is responsible for producing most of its hormones. Surrounding the infundibulum is a tubular extension called the pars tuberalis, which is highly vascularized and contains the portal vessels that connect the hypothalamus to the pituitary, allowing precise hormonal regulation. Finally, between the anterior and posterior lobes lies the pars intermedia, a thin zone containing colloid-filled follicles. In humans, this intermediate lobe is relatively small and underdeveloped, but it still contributes to hormone production. The pars distalis forms the largest portion of the anterior pituitary and is the main site of hormone production. Under the microscope, the secretory cells are arranged in cords and clusters around a network of sinusoidal capillaries, allowing hormones to enter the bloodstream efficiently. These hormones include growth hormone, prolactin, adrenocorticotropic hormone, thyroid-stimulating hormone, follicle-stimulating hormone, and luteinizing hormone. They are stored in cytoplasmic granules and released through a process called exocytosis whenever the body signals for them. Clinically, the pars distalis is particularly important because it is the most common site for pituitary adenomas, which are tumors that can disrupt hormone balance and cause systemic effects. The pars tuberalis is a thin, highly vascularized sleeve that surrounds the infundibular stem, the connection between the hypothalamus and the pituitary. It contains specialized cells called gonadotropes, which produce follicle-stimulating hormone and luteinizing hormone, key regulators of reproductive function. Interestingly, the pars tuberalis also contains receptors for melatonin, suggesting it may play a role in seasonal or rhythmic regulation of certain genes. In addition, it supports communication between the hypothalamus and the anterior pituitary through the hypophyseal portal vessels, ensuring precise hormonal control throughout the body. Functionally, the pars intermedia is less vascularized than the main anterior pituitary, which partly explains its more limited endocrine output in humans. Despite this, it still has physiological importance because it produces peptides derived from pro-opiomelanocortin, a precursor protein that is processed into several biologically active hormones. Among these are melanocyte-stimulating hormone, which plays a role in skin pigmentation and possibly appetite regulation, and beta-endorphin, which is involved in pain modulation and stress responses. Although the pars intermedia is small, these peptide products contribute to the broader neuroendocrine network linking stress, metabolism, and pigmentation pathways. Clinically, remnants of the pars intermedia can give rise to Rathke cleft cysts, which are benign cystic lesions that may become symptomatic if they enlarge and compress surrounding pituitary tissue. The posterior pituitary, also known as the neurohypophysis, is composed mainly of unmyelinated nerve fibers that originate from neurons in the supraoptic and paraventricular nuclei of the hypothalamus. Unlike the anterior pituitary, it does not produce hormones itself. Instead, it functions as a storage and release site for hormones synthesized in the hypothalamus. The two main hormones stored here are vasopressin, also called antidiuretic hormone, which is primarily produced in the supraoptic nucleus, and oxytocin, which is mainly produced in the paraventricular nucleus. These hormones are transported down the axons of hypothalamic neurons and stored in dilated axon terminals known as herring bodies until the body signals for their release. The posterior pituitary also contains specialized supportive cells called pituocytes. These are modified glial cells that help regulate hormone release by controlling the interaction between nerve terminals and nearby blood vessels. Surrounding the posterior pituitary is a rich capillary network that allows rapid entry of these hormones into the systemic circulation. This enables quick physiological responses, such as water balance regulation through vasopressin and uterine contraction and milk ejection through oxytocin. Clinically, damage to this region or disruption of vasopressin release can lead to disorders such as diabetes insipidus, highlighting the essential role of the posterior pituitary in maintaining fluid homeostasis. The anterior pituitary contains several specialized endocrine cell types that are traditionally classified based on their staining characteristics into acidophils, basophils, and chromophobes. Each group plays a distinct role in hormonal regulation throughout the body. Acidophils include somatotropes and lactotropes. Somatotropes are responsible for producing growth hormone and are typically found in the lateral regions of the anterior pituitary. Growth hormone plays a key role in linear growth, protein synthesis, and metabolic regulation. Lactotropes produce prolactin, which is mainly involved in milk production and breast development. These cells are more diffusely distributed throughout the anterior pituitary. Basophils include corticotropes, thyrotropes, and gonadotropes. Corticotropes produce adrenocorticotropic hormone, which stimulates the adrenal cortex to release cortisol during stress. Thyrotropes produce thyroid-stimulating hormone, which regulates thyroid gland activity and overall metabolic rate. These are the least common anterior pituitary cells. Gonadotropes produce luteinizing hormone and follicle-stimulating hormone, which regulate reproductive function, including ovulation, testosterone production, and gamete development. Chromophobes are cells that appear pale on staining because they contain little stored hormone. Many chromophobes are thought to represent resting or previously active cells that have released their hormone content. They may also include precursor or supporting cells within the anterior pituitary. Clinically, understanding these cell types is important because most pituitary adenomas arise from a single cell lineage, which explains why tumors often produce excess amounts of a specific hormone. The traditional view of the anterior pituitary is that each cell type produces a single specific hormone. However, newer molecular and immunohistochemical studies suggest that pituitary cells are more functionally flexible than previously thought. Some pituitary cells demonstrate multi-hormonal capability, meaning they can produce more than one hormone under certain physiological or pathological conditions. For example, a subset of thyrotrope lineage cells has been shown to co-express hormones such as thyroid-stimulating hormone, prolactin, or, less commonly, gonadotropins. This is thought to reflect shared developmental origins and overlapping transcription factor regulation during pituitary cell differentiation. In addition, pituitary cells may exhibit multi-responsive behavior. This means a single cell type can respond to more than one hypothalamic releasing signal. For instance, lactotrope and somatotrope activity can be influenced by multiple hypothalamic peptides, allowing the gland to adapt hormone output dynamically in response to physiological stress, metabolic demands, or reproductive state. Overall, this functional plasticity highlights that pituitary regulation is not purely linear or one-to-one. Instead, it is an integrated and adaptive endocrine network that allows fine-tuning of hormone secretion to maintain systemic homeostasis. Clinically, this concept helps explain mixed hormone-secreting pituitary adenomas and variable endocrine phenotypes seen in some pituitary disorders. The pituitary gland has a highly specialized blood supply that allows the hypothalamus to precisely control hormone release. The anterior pituitary mainly receives blood through the hypothalamic-hypophyseal portal system. In this system, blood first flows through capillaries near the hypothalamus, then travels through portal veins to the anterior pituitary. This low-pressure pathway allows hypothalamic-releasing hormones to directly regulate anterior pituitary hormone secretion. In contrast, the posterior pituitary is supplied primarily by direct arterial blood flow from the inferior hypophyseal arteries. This higher-pressure arterial supply supports rapid release of hormones that were synthesized in the hypothalamus and transported down axons into the posterior pituitary. Together, this dual vascular design allows tight regulatory control for the anterior pituitary and rapid systemic hormone delivery from the posterior pituitary. The arterial supply of the pituitary gland is uniquely organized to support both endocrine regulation and rapid hormone delivery. Two main arterial systems arise from branches of the internal carotid artery and divide functional roles between the anterior and posterior lobes. The anterior pituitary receives blood primarily from the superior hypophyseal arteries. These vessels typically arise from the supraclinoid or ophthalmic segment of the internal carotid artery, often between the ophthalmic and posterior communicating artery origins. They form a capillary network in the median eminence, which then connects to the anterior pituitary through the hypophyseal portal system. This low-pressure portal circulation allows hypothalamic-releasing hormones to directly regulate anterior pituitary hormone secretion without dilution in systemic circulation. In contrast, the posterior pituitary is supplied mainly by the inferior hypophyseal arteries, which most commonly arise from the meningo-hypophyseal trunk of the cavernous segment of the internal carotid artery. These arteries provide a more direct arterial blood supply to the posterior lobe, supporting rapid release of hypothalamic hormones such as vasopressin and oxytocin into systemic circulation. Overall, this dual vascular arrangement reflects functional specialization, portal vascular regulation for anterior pituitary hormone control, and direct arterial perfusion for rapid posterior pituitary hormone release. Venous drainage of the pituitary gland occurs primarily through the anterior and posterior hypophyseal veins, which empty into the cavernous sinus on either side of the sella turcica. From there, blood ultimately drains into the superior and inferior petrosal sinuses and then into the systemic venous circulation. There are important anatomical variations in this region. Transcellular vessels may be present, and there are often anastomoses between the superior and inferior hypophyseal arterial systems. These vascular connections contribute to the rich but complex blood supply of the pituitary gland. Clinically, this vascular arrangement is significant. Sudden hemorrhage or infarction within a pituitary adenoma can lead to pituitary apoplexy, a neurosurgical emergency characterized by acute headache, visual disturbance, and endocrine dysfunction. In addition, during transsphenoidal pituitary surgery, awareness of venous drainage patterns and arterial anastomoses is essential to minimize intraoperative bleeding and avoid injury to the cavernous sinus. As we finish, remember that the pituitary is more than just a small gland at the base of the brain. It's a command center for the body's hormones. The anterior lobe, or pars distalis, handles the bulk of hormone production, storing them in granules ready for release. The pars tuberalis wraps around the stalk and helps coordinate reproductive signals with daily and seasonal rhythms. The pars intermedia is tiny in humans but still contributes melanocyte-stimulating hormone and beta endorphins, while the posterior lobe, or pars nervosa, stores and releases hormones made by the hypothalamus, ensuring the body responds quickly to changes. Together, these lobes work as an integrated, flexible system, allowing the pituitary to regulate growth, metabolism, reproduction, stress, and water balance seamlessly. If you want to dive deeper into neuroscience and clinical insights, follow StudyNeuro on YouTube, Facebook, TikTok, Instagram, and Twitter at StudyNeuro. And remember to like, comment, share, and subscribe. We've got much more coming your way.